Well, a warm welcome to this talk. Now, I've just got off a rather long video call with Dr. David Speicher, the renowned Canadian virologist and analytical scientist. And I'm going to run through the main findings of what we talked about, and then you can watch the video in detail if you want all the nuance. So just a quick sketch really through what is we talked about on the video. Now, Dr. Speicher's analysed 32 vials of RNA vaccine so far, and has found DNA contamination in all of those vials that is analysed from different countries. And in the Pfizer vaccine is found something called SV, simian virus 40 enhancer and promoter. More on what that does in a minute, but basically it means that the DNA from the contamination can get into the nucleus of the cell and into the DNA of the host cell. The DNA is found is residual plasmid DNA from post-process impurity. So this DNA from E. coli bacteria, the sort of bacteria you've got in your colon, this impurity should all be taken out, but it's not. The manufacturing process has left it in Pfizer and in even larger amounts in the Moderna vaccines from the E. coli. Because viruses, of course, can't grow on their own. They can't make their own energy. So they've got to be cultivated in some sort of cell culture. And the cell culture used in the manufacturing processes is E. coli, that they're the sorts of cells, the bacterial cells, that the virus is brewed up in. There's free broken up DNA. So the DNA is partly broken up. So it's the E. coli are lies. They're broken up and the DNA and the RNA comes out. The RNA is what the manufacturers want, of course. But there's also DNA comes out as well. DNA fragments come out. But the thing is, this DNA is packaged into the lipid nanoparticles. So the DNA that's not supposed to be there is packaged into the lipid nanoparticles with the RNA, and that means it's systemically distributed around the body because these lipid nanoparticles go everywhere. That means the DNA from the post-processing impurity goes to the cells potentially all around the body. And that basically, the fact that it goes into the lipid nanoparticles guarantees that the DNA fragments get into cells. Now, the limits of DNA contamination that are allowed in vaccines were really formulated before the lipid nanoparticle technique was developed. So small amounts of DNA going into your arm won't do too much harm, but when that DNA is in lipid nanoparticles and it goes everywhere, it's a carrier mechanism to take it around the body. There's systemic biodistribution. And also that means that the lipid nanoparticle ensures the entry of the DNA contaminant into the body cells. So it really is quite a problem with guaranteed systemic distribution. Now, work from 1999 has shown that 3 to 10 copies of these DNA fragments, this SV fragment particularly, 3 to 10 copies getting into a cell can facilitate transport of that DNA contamination into the cell nucleus, as long as the cell is not dividing. If the cell's in what we call the interface, when there's no active cell division going on. So only 3 to 10 copies of DNA contamination can cause that contamination to go from the blood, from the lipid nanoparticles where the blood takes it to the cell, into the cytosol, the cytoplasm of the cell. And 3 to 10 copies means it can go into the nucleus of the cell. And once it's in the nucleus of the cell, what have we got in the nucleus of the cell? Well, we've got our own DNA, our own deoxyribonucleic acid. And it can be incorporated into that. When the foreign DNA gets into our own DNA, that is a change in our DNA. And when you have a change in the DNA or a change in the genetic material, that is defined as a mutation. And mutations can lead to malignant change. So integration would mean mutation. We know that that can happen. Now, cells in culture exposed to the lipid nanoparticles contaminated with the DNA. So human cells have been cultured, exposed to this contaminated vaccine, the RNA vaccine with the lipid nanoparticles. And when the host DNA of those cells has been analysed, it's been found that the DNA from the DNA that's contaminating the vaccine does get into the chromosomes of the human cells. Now, this has been demonstrated in cell culture. It's not been demonstrated in whole people, but there again, governments aren't looking for that. So let me just explain that a bit again. So they've made cultures of human cells. So you have a culture of human cells growing in a dish or something. You then add the vaccine, the mRNA vaccine, which contains the DNA contamination. You then let that brew for a period of time. You let it incubate for a period of time. Then you take the DNA from the host cells, human cells in culture. You take that DNA and the DNA that contaminated the RNA, the DNA, the contaminant that was in the lipid nanoparticles does get back into the DNA of the host cells. This has been demonstrated. So we know this happens in cultures, in tissue cultures. Now, what we need to do, of course, is take people, for example, who have long vaccine injury, take their cells, do a complete genomic analysis, and it will be a relatively simple matter to demonstrate whether this foreign DNA was in their cells or not, but that work is not being done. If you want to know why that work is not being done, oh, I don't know, ask a politician. They might know why it's not being done. So, and the chromosomes that have been contaminated, for want of a better word, the chromosomes are made of the DNA and some additional proteins called histone proteins. And chromosomes 9 and 12 have been demonstrated to have been contaminated with the DNA from the contamination from the lipid nanoparticles in the so-called RNA vaccines, which I guess we must now call RNA/DNA vaccines. And I've got a note here, cancer tumours a year post-vaccine. So basically there can be a delay before cancers form, I think is what I mean from that note. So the mutation's been proved in an oncogene. Now an oncogene is a gene which can stimulate mitosis. And of course cancer is an out of control mitosis. So we know that at least one oncogene, one potential cancer causing gene in chromosome 9 or 12 can have been altered by this DNA. So the theoretical risk is clear, and to me that theoretical risk is enough to stop these DNA contaminated vaccines until we know one heck of a lot more about it. But we know enough to stop it now unequivocally in my view. Health Canada has said there's no increased risk of cancers. And they also said at first that the Pfizer vaccine does not contain SV40, but they were wrong, it does. They later said that although the SV40 is present, so a bit of backpedaling there from Health Canada, that the SV40, the SV40 is the contamination sequence, one of the contamination sequences of DNA that can get back into the nuclear material of the cell and cause mutation. Health Canada says that's got no functional role. So first of all, they said it's not there. Then they said, oh, you know what, it is there. Then they said, but don't worry about it, it doesn't do anything. But then Health Canada asked Pfizer about the residual fragment. So having Health Canada declared that it does nothing, they then wrote to Pfizer. So Health Canada know that this contaminating SV40 potential cancer-causing sequence is there. And then Health Canada was asked, like a Freedom of Information request, Health Canada was asked for this information from Pfizer, but it came back redacted. Why would they want to hide the scientific information, Health Canada? Why would they want to do that? Now, Dr. Speicher uses a technique called fluorometry. Now, this basically attaches a fluorescent molecule to the DNA. And then that means when the DNA is present, you can see it with your microscope. It fluoresces and you can actually see it. It's giving off light. Now, this is a good technique. This fluorometry is a good technique to give quantitative analysis of all of the DNA. So it analyzes all of the DNA that's present. It's a better technique than quantitative PCR because it's analysing all of the DNA and labelling with this fluorescent marker all of the DNA that's present. And it's found that in some DNA, in some RNA vaccines, there is 10 trillion copies of this DNA sequence per dose. Now, I was taken aback by this. Dr. Speicher has found that the Moderna one particularly, can contain up to 10 million copies of DNA fragment per dose. And we've just said that 3 to 10 copies of SV40 DNA fragment is enough to facilitate transport into the person's own DNA, into the cellular nuclear DNA. So 3 to 10 copies per cell and up to 10 trillion copies. So that means that potentially 1 trillion, that's 1,000 billion body cells, could be transfected with foreign DNA. And that just, I was taken aback by that. So 10 trillion copies of DNA contamination potentially present per dose, 3 to 10 copies enough to cause incorporation of SV40 DNA contamination into the nuclear genome of the cell, thereby causing a mutation which has been identified in cell cultures in chromosomes 9 and 12, including an oncogene that can potentially cause cancer. 10 trillion copies per dose, or up to that. So the DNA is likely to go into the nucleus. Now, I then asked if there's any possibility of germ cell contamination. So in other words, in the ovaries and testes, you've got cells which are giving rise to the next generation of sperm and ova, which gives rise to the next generation of human being. Now, the key thing here is if there's contamination, if there's contamination in a germ cell, that can cause a mutation in the germ cell, and that genetic mistake, if you like, that mutation can be passed on to the next generation. Therefore, you could get potentially genetic information to make spike protein passed on to the next generation. Theoretical possibility. Doesn't happen. Does it happen? We don't know. No one's bothered testing. I would have thought that should be #1 priority for governments around the world and authorising agencies around the world. But as far as Dr. Speicher is concerned, he doesn't know that it's been done. So I think we can probably assume that it hasn't been done, unless it's been done and we haven't been told about it. Maybe that's what's in all these black bits that are marked out in these redacted papers. Maybe that information is only for clever people, not for hoi polloi like you and me and Dr. David Speicher. But of course we don't know. We're speculating because we don't know if the work's been done or not. But a theoretical possibility. But let's hope not, he says, that we don't want ongoing spike production in the next generation. But of course this can explain long COVID. So long COVID could be explained by the fact that the DNA information to make the spike protein, for example, has got into the nucleus of the cell. Once that genetic information is in the nucleus of the cell, that can undergo a process called transcription, forming messenger RNA. That can then go to the ribosomes of the cell, a process called translation, where it will make the spike protein. Now, David Speicher at the moment, David is working on a test to test for the presence of ongoing production of spike protein. And he's developing a test that will test for the presence of spike protein in blood and in urine. So vital work being done there. Then if someone had long COVID, we could just test the blood, test the urine and say, you know what, you've got ongoing spike protein. And that would tell us, potentially, what is causing their symptoms. Whatever it's causing or not causing, it shouldn't be there. It's an abnormality and must be accounted for. So modified, now the other thing is that the RNA that's used is modified. One of the bases is modified, and that makes the RNA last for longer. So we've got this potential problem of the DNA contamination, but we've also got the problem of this modified DNA. Now, I asked him how long modified RNA. So I asked him how long the RNA would last for, potentially inside the cytoplasm of the cell, to give rise to, say, an abnormal spike protein. Because I would have thought it wouldn't be long, but he said it could be several months, and I was surprised. But he said this is because it's modified. One of the bases is modified to make it live for longer, to make it last longer. So people with long COVID that are having high levels of spike protein for long periods of time, at least for several months, could be explained by the fact that the modified RNA is hanging around inside the cell cytoplasm. It could also be explained by the fact that the DNA from the DNA contamination in the vaccines, that DNA has got into the cytosol of the cell and back into the nuclear material of the cell, and is actually become part of the cell in its ongoing processes of transcription and translation are continuing to produce it, and that could potentially go on forever because it's become part of the cell's DNA, which is quite a frightening prospect. He also pointed out the possibility for frame shifting. Now, the way this works is the genetic code is read, so 3 bases relate to one amino acid, and then the next amino acid and the next amino acid till you end up with a long string of amino acids. Then they then fold into more complex structures. Quite amazing process, protein folding, to give rise to 3-dimensional structures, which are the functional proteins of the body. But if that is moved along one, then the code can the code will still code for the same amino acid, but if it's moved along one, it could be coding for a different amino acid. So we need 3 bases, what's called a codon, to code for one amino acid. But if that shifted along one, then you'll get 3 different bases, and that could code for a different amino acid. So what this means is we'll get a range of proteins being produced. So I asked David about this, and he says, yes, this is true. And this frame shifting could result in the production of short protein sequences of part of the spike protein, or medium length proteins of part of the spike protein, or long lengths of the protein causing spike protein. So in the centre, we've got a polyclonal ongoing potential production of spike protein, and that could cause ongoing inflammation, which could account for some of the problems that we're seeing in patients with long COVID. There could be many other causes, of course, but that's potentially one of them that's theoretically a possibility. And also, I learned a lot during this talk. He says there's possibilities for double-stranded RNA. I'd never heard of that, but he says it's possibilities for that occurring. So well, I accept it. And he also said there's possibilities for DNA, RNA forming complexes together. Again, I'd never heard of that, but strange stuff, but doesn't sound good. It's not normal, as I understand it, and therefore we should put a moratorium on RNA vaccines until we get this science clarified. Safety for the vaccines has not been shown, but we do know that they cause harm. He pointed out that Pfizer did use a gene therapy plasmid to develop as part of the vaccine development. David Speicher congratulated Port Hedland Council for raising the alarm about this, and we talked about this in the last video with Russell Broadbent, the Australian Member of Parliament. So Port Hedland has alerted its doctors in its jurisdiction to this risk. So the doctors should be talking to their patients about this. That means the patients can give informed consent. And also Port Hedland Council is communicating with, I think it was the 550 other councils in Australia, to alert them to the danger. So good courage demonstrated by Port Hedland Council. We then talked about genomic analysis, that the fact that we could be looking at the DNA. So people that have been vaccinated, especially those suffering from vaccine damage, it would be a relatively simple matter. We could take some tissue or even potentially some blood, do a full genomic analysis on that, and identify if this pathological this pathological DNA from the DNA contamination in the vaccines has in fact been incorporated into living human cells. I asked why this hasn't been done, and basically the answer is governments don't seem interested in doing this sort of research. I would have thought it should be their top priority, but what do I know about it? Right, government's spike protein test would work out the government. Our governments aren't doing this, but David's working on a test to look for the spike protein. So David's test is not looking for the DNA contamination. That needs a genomic analysis test. But David is working on a test that will test for the product of the abnormal gene that's now been incorporated into our DNA, and is resulting in the production of abnormal spike protein in the blood and urine, if indeed it is there. That's what his test will show. So absolutely brilliant that he's doing that, but that should be combined with research on full gene sequences from living human beings, or indeed from post-mortem material from dead human beings who may have died in circumstances which are unclear, but that is currently not being done according to the best of his knowledge or my knowledge. And also quite incredible that David is not financially supported to do this work, so there will be a link to support his work below the video. And then we just closed, he's also starting to do some work on the replicon vaccine launched in Japan, which is frightening for him and for me, but I don't know much about it, so I'm not going to talk about it now, but that will be the subject of future videos. When will human beings realise it's time to stop tinkering with things they don't understand? And when will human beings learn that making money is not the top priority of human existence? Let's now listen to the full interview with David. I might put this on as a separate video, but we'll put both up. But quite alarming things to think about, really. So on to David now, and thank you, David Speicher, for the work you've done and for taking part in this interview.